Welcome everyone. Today we are going to learn about ATP-driven pump. Now, at first I want to mention that there are three major categories of transport protein is present. One is channels, second is transporter. Under the transporter there are uniporters, symporters and antiporters. And the third category is ATP-driven pump. Now, ATP-driven pump contains four different class of proteins. One is called P class pump, V class pump, F class pump and ABC transporter. Now, in this video, I'll be going to talk about P class pump. Now, before I begin to talk about P class pump, I want to mention some basic facts about ATP-driven pump at first. The first point, we have to understand that through the ATP-driven pump, active transport actually occur. Active transport occur. Now, why the transport that occurs through ATP-driven pump is called active transport? Because in this active transport, in this pump, energy is required. Energy is required. Now, in my previous video, I have mentioned that there is two types of movement occurs during transport. One is called downhill movement and another is called uphill movement. These two points are so important that I have mentioned, I think I have mentioned in all my transport videos. Suppose this is an example of, this is the example of hill. So what we know that if someone try to come down from the hill, that's called downhill movement. It requires less energy, less effort, so it feels easy. But if someone try to climb up the hill, so we call uphill movement, it requires more energy and more effort. The same is true for molecules. When molecules move from higher to lower concentration, it is called passive transport and it does not require energy. But when molecules move from lower to higher concentrations, like the uphill movement, during uphill movement, it requires energy. That's why it's called active transport. Now, second point is here energy is actually required, and in case of ATP-driven pump, the hydrolysis of ATP generates the energy. Now, this ATP-driven pump can also be called ATPage, but this ATP-driven pump do not hydrolyze ATP when they are not transporting ions. So, ATP hydrolysis and the transport of ions are actually coupled. So when transport of ions occur, then only the ATP will be hydrolyzed. When there is no transport occurring through this pump, there will be no ATP hydrolysis. So this fact we have to understand at first. Okay, so active transport occur, energy required, ATP hydrolysis occur, and these two things, ATP hydrolysis and transport, are coupled. Now I will talk about the P class pump. This structure is actually the basic structure of all P class pump. P class pump. Now, this domain of the protein is called transmembrane domain of the P class pump. Transmembrane domain. This domain actually spans the lipid bilayer. Now, this three other segments is called, this is called A or actuator domain. Actuator or activated domain. This is called P domain or phosphorylation domain. And this is called N domain, that means nucleotide domain. Nucleotide binding domain. Now, I want to mention here why this is called P class pump, because of these phosphorylations. Here, all this P class pump contains these four different segments, but this phosphorylation is must. In this phosphorylation domain, there is a particular amino acid present called aspartic acid. Remember this, aspartic acid. Now, in the nucleotide binding domain, ATP will bind, and the ATP actually contains adenosine and triple phosphate. We know that. This phosphate is called alpha, this is the beta, and this is the gamma. This gamma phosphate is the terminal phosphate, and this phosphate will break down to make ATP and the inorganic phosphate. Now this phosphate will bind with this aspartate or aspartic acid amino acid that is very conserved. Remember this, it is highly conserved. Highly conserved in all the P class pump. That's why it is called P for phosphorylations. Okay. So in this P domain, there will be an aspartate, and that aspartate will be phosphorylated. So here a phosphate group will be attached. Now when phosphorylation occurs, then only this pump can transport ions. Okay, so this is the very basic structure of all the pumps that I have drawn here that actually work in different cells. But the basic structure will be this. There is an activated domain, there is a phosphorylation domain, there is a nucleotide binding domain where ATP bind, and this breakdown of this terminal ATP will actually bind with the aspartate, and then a rotation of the pump occurs. Through this, the ions will move from outside or it can come inside. So now let's talk about different pumps present in different cells, how it works. As you can see, this hydrogen pump is present in plant and fungi. Through this hydrogen pump, hydrogen ions are actually pumped out of the cell of plants and fungi to generate H plus gradient. H plus gradient. Now what is H plus gradient or proton gradient? That means there will be a difference in the concentration of hydrogen than outside and inside. Suppose this is the exterior of the cell, and this is the cytosol. So there will be a high concentration gradient of hydrogen ions over the exterior and the cytosol. So this pump actually generates the hydrogen or proton gradient. So it's found in the plant and fungi. Here H plus pumps, here also ATP breakdown occur and the phosphorylation occur of aspartate acids. Okay. Now this is called sodium potassium pump. This is very important because all animal cells have this pump. All animal cells. In the plasma membrane of all animal cells contains the sodium potassium pump. What happens here? 3 sodium actually pump out of the cell and 2 potassium import occur or enter the cell. Now what happens here? As you can see, this pump actually export sodium ions from inside to the outside. So the concentration of sodium will be high outside and will be low inside. So this sodium potassium pump actually generates the ion concentration gradient of sodium and potassium. Basically, all ATP driven pumps generate and maintain the ion concentration gradients. So by this functioning of this pump, the sodium ion and the potassium ions concentration gradient will be generated. In all animal cells, the outside of the sodium concentration is high. It is approximately 145 millimolar and inside is 12 millimolar. The opposite is true, as because you see that potassium ions move inside, so there will be low concentration of potassium outside and high concentration of potassium inside. It has been found that there is 155 millimolar concentration of potassium inside and outside is only 4 millimolar. Now one thing I have to mention that this pump is actually electrogenic. Remember the term electrogenic. Why it is called electrogenic? Because it pumps 3 positive ions out and 2 positive ions in. So here 1 positive ions will be deficient inside the cell, so the inside of the membrane becomes negatively charged. By the working of this pump, the inside of the membrane starts to become negative charge because more positive ions are moving outside than it is coming inside, and more positive charge will be present outside. Now one point I have mentioned in my previous all videos, that there is two driving force work for the movement of ions. One is called chemical concentration gradient and called membrane potential. These two factors work together for ions and it is called electrochemical gradient. Electrochemical gradient. Okay. Now, sodium potassium pump actually contributes this charge separation, that is called the membrane potential, only 10%. One channel is present in all animal cells called non-gated potassium leaky channel, or we say this channel as potassium inward rectifier. You can simply remember rectifier. You can simply remember potassium leaky channels. So these channels actually contribute the major membrane potential during the resting phase of the cells. What happens here? Potassium ions are actually exported from these channels, as because I have mentioned that potassium ions are higher in concentration inside. So this pump actually moves the potassium ions from inside to the outside, and by this way it actually generates the resting membrane potentials, and this pump actually contributes approximate 90% of this membrane potential, that is called resting membrane potential. The voltage difference is here minus 70 millivolt. Remember, this is the generally the resting membrane potential. Now this is not today's topic, but I have mentioned because of the generation of resting membrane potential, these two factor works. The sodium potassium pump only contribute 10% of the membrane potential, and the 90% is actually generated by the potassium leaky channels. Now in the apical membrane of stomach, apical membrane of mammalian stomach, basically there is a cell called parietal cell or oxyntic cells that secrete hydrogen ions outside the cell in the stomach lumen. So one hydrogen export occur and one potassium import occur. Okay. This pump is also under the P class pump. So that means here phosphorylation occur and ATP breakdown occur. So in all this pump, there will be a breakdown of ATP and the phosphorylation of a aspartate acid or amino acid in a P domain. So this pump actually works to develop the low pH in the stomach lumen so that the food can be digested, pathogen can be killed, and some enzymes like pepsins, these proteolytic enzymes, works at the lower pH. So I will make a separate video on H plus and potassium pump, how the stomach oxyntic or parietal cells actually generate this the low pH. I will make a separate video on this. Now under the P class pump, there is also some calcium pumps present. In these calcium pumps, calciums are transported out of the cells, that means calcium export occur, and simultaneously hydrogen import occur. This calcium pump is called plasma membrane calcium ATPase. Now why it is called ATPase? Because it breaks down the ATP. That's why it is called ATPase. So one calcium comes out, one hydrogen comes in. In the SR means here sarcoplasmic reticulum. Sarcoplasmic reticulum. These intracellular organelles are present only in the muscle cells. It could be skeletal muscle or it could be cardiac muscle. In the SR, there is a calcium pump also present. Just like the plasma membrane calcium pump, there is a calcium pump present in the SR. That's called SARCA, ACRCA. And there in the skeletal muscle, the SARCA is called SARCA1. In the cardiac muscle, it's called SARCA2. Now, one very important point I can mention here, that this sarcoendoplasmic reticulum contains approximate 80% of this SARCA calcium pump in the membrane. So you can understand how much this pump is important. Now I will talk about what is the role or significance of this calcium pump. Now what happens here, in this through the Sarka pump, calcium are actually goes inside the sarcoplasmic reticulum. Now, one thing I have mentioned in my previous video, where I have talked about how muscle contraction occurs, I mentioned that when the calcium concentration increases, contraction occurs. Okay, and when the calcium concentration falls, what happens? Relaxation occurs. The calcium concentration inside the cell is tightly controlled. Inside the cell, the calcium concentration is 10 to the power minus 7 molar. But in the sarcoplasmic reticulum, the calcium concentration is minus 10 to the power minus 3 molar. That means there is 10,000 fold difference. And also it is true for outside of the cell, there is 10 to the power minus 3 molar calcium concentration. So outside of the cell and the sarcoplasmic reticulum contains huge amount of calcium ions. But in the cytosol, only 10 to the power minus 7 molar calcium present. That means there is a 10,000 fold difference. Now, by removing this calcium from the cytosol to the sarcoplasmic reticulum or through the outside, it actually lowers the calcium concentrations. That means this pump actually helps in relaxations. Remember this. This pump helps in relaxations. Now, how contraction occurs? So which transporter proteins are actually responsible for contractions? Two important channels are actually responsible for the contractions. One is called this voltage gated calcium. In case of heart muscles, there is a particular calcium channel present called voltage gated calcium 1.2. Through these channels, basically calcium import occurs. As because outside concentration of calcium is high than inside, so calcium will move from outside to the inside. But through the calcium voltage gated calcium channels, in case of skeletal muscles and heart muscles, small amount of calcium actually released. The major amount of calcium are released through the sarcoplasmic reticulum, and this is called ryanodine receptor. Ryanodine receptor. Basically, they are actually channel. Okay. Now, why it is called ryanodine? Because a plant alkaloid called ryanodine can block the channel, so it's called ryanodine receptor channel. But through this channel, actually calcium export occur. So calcium will come out through the channels. So huge amount of calcium is actually export occur inside the cytosol, and through this Sarka 1 or Sarka 2, in case of heart it is called Sarka 2, in case of cardiac muscle we call Sarka 1, this calcium actually come back inside the sarcoplasmic reticulum. So through these channels, calcium concentration increases and contraction occur. After the contractions, through the Sarka 1 and through the PMCA, calcium export occur and relaxation occur. Now, in case of, I want to mention here that in case of cardiac muscles, this ryanodine is called ryanodine 2, and in case of skeletal muscles, this is called ryanodine 1 receptor. Now, also one antipoda helps in the relaxation, that is called sodium calcium antipoda. I have also mentioned here. Basically, 3 sodium actually comes inside the cells, and 1 calcium actually export occur. 1 calcium actually removes through the antipoda. So by this PMCA, this sodium calcium antipoda and the Sarka 1, relaxation occur. Through the voltage gated calcium channels and ryanodine receptor channels, the contraction occurs. Hope this video will help you. Thank you for watching, and always keep learning.